Alrighty. Kia ora, everyone, and thank you for joining us today for our digital event, the final of our Brain Awareness Month series. It's a very exciting day today. Before we kick off with Dr. Mugisho, I'm just going to run through some housekeeping slides for those who are new to our events. So please just pay attention while we go through these. So we like to kick off our events with a whakatauki, and this one here you can see sits above the desk of our head of research, Dr. Sarah Schomburger. So it goes ehara, takutoa, toa, iti toa, takitahi, engare, he toa, takitini. And that translates to my strength is not as an individual, but as a collective, which we think is very appropriate in the research space. So as you can see, your camera and microphone have been automatically turned off, and you won't be able to turn these on. This means we cannot see or hear you, and it will remain that way for the duration of the event. But if you do need to communicate with us, you can use the chat and Q&A functions at the bottom of the screen. So if you just look down the bottom of your screen, you'll see the icons there. You click on chat and it will open up a box, a small pop-up window where you can chat to myself. No other attendees will be able to see what you write in this box, so you can use this for any technical questions that you might have, and I will do my best to help you. If you have any questions you'd like to ask about today's topic, particularly at the end when we open things up for a Q&A, you can use the Q&A function. So same thing again, the icon down the bottom of your screen, click on this and it will open up a new pop-up window. So you can type your question in here. Please note that your questions will be visible by everybody else. However, if you would like to, you can tick this little box here and you can send it anonymously. Once it is submitted, we will either opt to answer it live or we might type a response to you. So just keep an eye on that. And please bear in mind that we have a limited time frame today, so we might not be able to get to all of your questions, but we will do our best. And please do keep all of your questions related to the topic presented. Our speaker won't be able to answer any personal medical related questions that you might have. At the conclusion of the session, it will end for everyone, so apologies in advance, it can be a little bit abrupt. But the recording of this will be available on our website at a later date. So please bear in mind this is being recorded. Okay, on to our lovely speaker for the day. So we have with us today Dr. Lola Mugisho, who is a senior research fellow at Auckland University. And that's also where Dr. Mugisho got her PhD. She is Deputy Director of the Buchanan Ocular Therapeutics Unit at the University of Auckland and is the recipient of several prestigious grants and awards, including the Health Research Council Emerging Researcher Grant and the New Horizons Women's Trust Margaret L. Bailey Science Award. Dr. Mugisho's research focuses primarily on the role of the inflammasome pathway in neurodegenerative diseases. So please join me in welcoming Dr. Mugisho, and I will now hand on over to you.
Oh, thank you. Thanks, Juliet. Just a moment. I'll just share my screen. Just a moment, sorry. Okay. Just a moment. Again. Cool. So can everyone see my presentation? I guess you can respond, but Juliet, you can.
I can't see it, Lola, sorry.
So this is on 2. Yeah, okay, just a moment. I'll try that again. Let me try that again. Now?
No, unfortunately, we can't. It's very strange.
For
For all those at home, we did run a test.
Yeah. Okay, let's try that again.
Here
Now?
we go. Yes.
Okay, perfect.
Fantastic.
I don't know what happened there. Okay, sorry, just a moment. Okay, I'll just get into it. So hi everyone, my name is Lola. As Juliet said, I'm a senior research fellow in the Department of Ophthalmology at the University of Auckland. I'm also the Deputy Director of the Buchanan Ocular Therapeutics Unit. And this unit also sits within the Department of Ophthalmology. I've put my email address there just in case anyone has any additional questions that they want to ask me, to email me directly, and I'll try to get back as soon as I can. Anyway, for today, my talk is on the role of inflammation in neurodegenerative diseases. And the way the talk is structured is such that we will kind of follow my research journey from my master's through to now, and then overlay that with how the research has developed, and sort of some of the cool things we've been able to do over the years, the last 10 years or so. Okay, so let's start with inflammation. What is inflammation? So inflammation is the body's natural response to harmful stimuli. So this could be infections, it could be irritants, it could be injury. And usually I'll just see if I can use a pointer, just a moment. There's a pointer. Yeah, so usually it's a complex process, but it involves the recruitment of what we know as immune cells, of different immune molecules, and also of different pathways. Inflammation can either be acute or chronic. So if you have acute inflammation, it's like, let's say you get a cold, and then your body triggers all of these pathways to help you fight your cold symptoms, and then everything resolves and you're back to normal. That's an acute response. So it's a good thing. An acute response is necessary for the body to heal. But then sometimes inflammation can be chronic, and this is when it becomes a bad thing. It's when, unfortunately, that inflammatory pathway that we've triggered does not shut down itself, and it keeps going and starts to cause damage to our body. And most of the time, and this is the type of the inflammation that is linked to neurodegenerative diseases. This neurodegenerative type of inflammation, this chronic inflammation, is particularly evident with age. And with age, we see an imbalance between anti-inflammatory. So anti-inflammatory are the good things for you. They're supposed to shut down that inflammatory pathway. And pro-inflammatory, which is the bad side. So you have more pro-inflammatory pathways activated, molecules released, and not as much anti-inflammatory. And like I said earlier, neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, are all linked to chronic inflammation. So why study inflammation? Well, my first introduction to inflammation actually started in my master's, which I completed at the Center for Brain Research here at the University of Auckland. And I was supervised by both Dr. Simon O'Carroll and Emeritus Professor Lewis Nicholson. And the question we were trying to answer during my master's was, where does the inflammation that affects the brain in neurodegenerative diseases come from? The reason we're looking at this question is because we know that when you have diseases outside of the brain, like in your heart, in the lungs, and the kidneys, they all release these inflammatory molecules into the blood system. So we wanted to know that, do these inflammatory molecules released into the blood system go into the sensitive blood vessels that are in the brain and damage them, cause them to be more leaky? When they become more leaky, you have a lot of those toxins being able to leak into the brain and then potentially contribute to brain diseases. So pretty much what we were trying to see is, can peripheral diseases, which are diseases outside of the brain, make us more susceptible to brain diseases? So to do this, one of many experiments that we did was to take the cells in the blood of the blood vessels, they're called endothelial cells. So we took these endothelial cells and we cultured them in what's known as an insert. So what you do is that you put them at the base of the insert and they grow into a nice single layer of cells, and they form a barrier. And so you put some nutrient at the top and some nutrient at the bottom. The nutrient here is in pink. And then you can add in, inflammatory agents like bradykinin. So in our study, we use bradykinin because it's a well-known inflammatory molecule that is released when you have inflammation throughout your body. So we add it into the cell layer at the top, and then we can measure how leaky that cell layer gets. So if there is, so what we do is we add some dye at the top, and then we measure how much dye gets leaked to the bottom. So if there's inflammation, and we wanted to see if there's inflammation, do we have weaker connection between the cells? So the cells become more leaky, so more dye goes through to the bottom. And this is what we found. So when we added the bradykinin in the form of desH9-BK, we found that the cell layer was more leaky compared to if we added nothing at all. So this tells us that the inflammation or bradykinin, which comes from the outside of the brain, can actually affect the blood vessels within the brain and perhaps cause the leakage of toxins into the brain, contributing to brain diseases. And this was really where I got interested in inflammation-mediated changes to our blood vessels or vascular pathology. And very interesting is the fact that even though we see this leakage of blood vessels happening in the brain, it doesn't happen in the brain only. It also happens in other organ systems like the eyes, the kidneys, and the heart. But out of all of these structures, the eye is actually really relevant and important to look at because it gives us a unique opportunity to study the effect of inflammation on blood vessels, because it's the only one that we can see without invasively, without any invasive techniques. So for the brain, we can't just have a look at the blood vessels, but with the eyes, you can actually look through your eyes and see the blood vessels at the back of the eye. So particularly the structure we're interested in is right at the back of the eye called the retina. And it's a light sensitive structure that collects all the light impulses and converts it to electrical signals that are then sent to the brain. So a lot of times we look at the retina as a window to the brain because it is technically an extension of the brain, and it has neuronal cells and astrocytic support cells just like the brain does. So the retina allows us to observe these blood vessels and how they change in real time in diseases versus in normal, and therefore allows us also to test any new therapies. So because of this, I then for my PhD went across to the Department of Ophthalmology, supervised mainly by Ilva, and then on the rest of my supervisory team, my co-supervisors were Colin G and Monica. So it was a big team of researchers, and together we really wanted to know how inflammation affects the blood vessels in a disease known as diabetic retinopathy, and I'll talk about it in a minute. And then whether or not we can treat it. Are you not moving? So diabetic retinopathy is a chronic vascular disease of the retina. So it's a disease that affects that retinal structure that I spoke about and affects the way we see. So when we look at the blood vessels, so this is in real, through the lens, through the cornea, going through the lens, looking at the retina at the back of the eye, you see this beautiful vascular network here in white. just like a nice tree diagram, really, spreading across what is known as the fundus. And this is in a normal eye. So this is what it should look like. But in diabetic retinopathy, it's a completely different picture. You have these areas of intense white here that actually indicates the area of the vessel leaking, which means that the vessels have lost their integrity. You have little microaneurysms here, being the little spotty white spots, and then you've got hemorrhages, these bigger ones being hemorrhages. And even some of the bigger vessels are a bit more tortuous, so they're a bit more windy. And so diabetic retinopathy is a largely vascular disease, and the vessels are pretty damaged in the disease. So what does seeing look like? Well, patients with severe diabetic retinopathy don't see a whole lot, unfortunately. And so it's the leading cause of vision loss in the working age population. Unfortunately, though we have current treatments that can help to reduce the the signs of the disease in later stages, the treatments don't actually prevent the development of the disease, and it doesn't stop the progression of the disease. So we need alternative treatments. So to start to answer our question about treating DR, the first thing we did was to look at the best models to use. But unfortunately, what we came across was that a lot of the models were focused on diabetes only. So like I said, diabetic retinopathy is a disease that affects the retina, but it is actually caused mainly by diabetes, which is where you have high blood glucose or hyperglycemia. However, it is not just hyperglycemia alone. It is associated with chronic inflammation. So having models that only show you hyperglycemia alone doesn't fully recapitulate what the disease should look like. So we first of all set out to prove that you need a combination of both hyperglycemia and inflammation together to cause the blood vessel leak and disease that we see in the disease. In diabetic retinopathy. So working with the New Zealand National Eye Centre and the eye bank affiliated with this eye centre, we were very fortunate to get our hands on some human donor tissues. And so we had human donor tissues from normal, by normal, I mean no known ocular pathology. And then we had donor retinas from patients that had confirmed diabetic retinopathy diagnosis. And we took sections through their retina, and then we did immunostaining on the retina. So pretty much staining for proteins. And I'll get you to ignore the green for now and focus on the red. So the red here is a marker of how inflamed the retina is. And you can see that when you have no known ocular pathology, there's barely any inflammation in their retina. But then when you come to the patients with confirmed DR, you have a lot more red, which means you have a lot more inflammation and the retina is really unhappy here. So what we wanted to do was to show that in order to attain this level of inflammation, you need both hyperglycemia and inflammation together. So we collaborated with a group at the Lions Eye Institute in Perth, Australia, headed by Professor Elizabeth Rakoshe. And we were lucky to get some Akita mouse tissues. And Akita is a hyperglycemia only mouse model. And we also got an Akimba mouse tissue where the model contained both hyperglycemia and inflammation together. And so what we wanted to do was compare the hyperglycemia and inflammation versus hyperglycemia alone versus normal, and see which of these disease conditions actually replicates what we see in diabetic retinopathy. So again, focusing on the red, you can see that in the normal tissues and the Akita tissues, there really is no difference in the level of red you see, and the red levels are pretty normal. But then when you come to akimbo, where you've got the hyperglycemia and inflammation together, you start to see a lot of red staining that really looks like what you see in diabetic retinopathy donor tissues. And when we quantify it, we can see that obviously the akimbo mouse has the higher level of the red staining compared to the Akita and the normal tissues. Now, the inflammation pathway is broad and it's big, and there's so many different pathways incorporated within it. But through our research, we found that the main pathway that plays a role in chronic diseases was one of the main pathways is the inflammasome pathway. Now, the inflammasome pathway is known as a pathway that is part of the innate immune system. Now, this immune system is very important because it's usually the first line of defense. So when you have any inflammatory agent come into your body, if you have an infection, an injury, the first system to respond is usually the inflammasome system. Now, the response can be acute, which is a good thing. But when we have, especially in age-related diseases, it can actually become chronic and has been shown to be a part of several chronic diseases in our body. The reason the inflammasome pathway it can be bad for us is because it can trigger this vicious cycle of chronic inflammation where we have a danger signal, whatever it might be in our system, that activates the inflammasome pathway. Then that pathway really causes release of pro-inflammatory cytokines, which can result in cell death, and then that re-triggers the whole pathway, and the cycle just keeps going round and round and round and worsening with time. So what we wanted to do was to see, okay, now we know that hyperglycemia and inflammation together is required to trigger, to mimic diabetic retinopathy. Can we block this by targeting the inflammasome pathway? So we go back and we did a whole host of experiments, but I'm going to show you the results based on the same model that I talked about earlier, which is where we grow the cells in an insert. But this time we added into that insert high glucose and inflammation alone in the first instance to trigger that leakage that we saw last time. Or we add it with a drug called peptide 5. And now peptide 5 is a drug developed here in New Zealand by Professor Colin Green and his team. And it's a known inflammasome pathway inhibitor. So we want to see, can we trigger that leakage of the cells, and can we recover it or protect with peptide 5? And just showing you B first, which is the dye-leak results, you can see that with the injury, we have increased leakage of the dye, and then peptide 5 actually prevents it. The other cool thing that we can do with this technique is that we can actually measure the electrical resistance across that layer of cell by having an electrode in the inner chamber and another one in the outer chamber, and then measuring the resistance across it. And when we do, with the injury, we see a decrease in resistance, which means that the cells are leakier because ions can flow through easier. And then with treatment, we see that we can recover, here in the blue bars, we can recover some of that electrical resistance. So the cell layer is tightening back up again, which is really interesting and cool. So next, we tried to do the same thing in vivo in the animal model. And we found that with our injury alone, you see this increased level of red, like what I showed you earlier, the red staining, which means the retina is really inflamed and it's unhappy. But then we don't see as much red with the peptide treatment group. In fact, it's looking more like the control where it doesn't have any injury at all. But what does this mean for leakage? Because this is all on a molecular level. What does it mean in terms of whether or not the vessels have integrity in this model? Well, there are two ways that we can look at vascular integrity. And this is based off a technique known as optical coherence tomography. I'm not going to go into it, but pretty much what it is they're taking a section through the retina. So this is the whole retina there, and that is the optic nerve head. And we can see pathologies within that retina. So for example, what you see here is known as a hyperreflective foci, and it's pretty much an indication that we have inflammation and cell death happening in the retina. When you have spaces that look like this, it means that you've got fluid buildup in the retina because you've got leakage happening. So what we found is that when we treat with peptide 5, these areas of inflammation and cell death were reduced by peptide 5 treatment. And this area of leakage was also significantly reduced by peptide 5 treatment. So essentially what this result shows us is that peptide 5 can protect, and by blocking the inflammasome pathway, can protect against both cell death and also vessel leak. But then when we go back to current treatment of DR, they have two key limitations. One is that they target downstream of the disease, which is why they don't affect the progression of the disease or the development of the disease. And by targeting upstream, we now show that we can actually address this limitation. But there's another limitation of this drug, of current drugs, and that is that they require intravitreal injection, which, as you can see in this picture, looks incredibly unpleasant. But unfortunately, peptide 5 so far needs to also be given intravitreally. So can we actually find another way to address this particular limitation? And this was the aim of my first postdoc. So after completing my PhD, I worked again with Ilva, Colin, and Monica on an AMRF-funded project where we asked the question, can an orally delivered inflammasome blocker protect against diabetic retinopathy? Interestingly, we had developed and characterized a drug in our group called tonabacet, which is a small molecule benzopyrin. So if you're interested in chemistry, I've got the structure there for you. But this molecule was initially developed by GSK several years ago. And GSK had actually taken it through phase 1 clinical trials, which means it's gone through all the safety tests. But unfortunately, they didn't know how it worked. So we came back later on and then showed that it actually blocks the inflammasome pathway. And now it's going through phase 2 clinical trials for diabetic retinopathy, which is the first trial stage where they look at the effectiveness of the drug. But before we go into looking at whether tonabacet protect against diabetic retinopathy signs, it was really important for us to check whether it works in the same way as peptide 5. And so with my technician at the time, the lovely Heather, we did some cell culture work. Again, a whole host of experiments, but we'll just show you what happened with the barrier integrity studies. But this time, instead of peptide 5, we used tonabacet. And we showed that if we look at that electrical resistance, the injury alone again decreases the resistance, but tonabacet was really good at protecting against this loss of electrical resistance across a single layer of the cells. When we look at dye leak, we see increase in dye leak with the injury, but again, tonabacet was really good at protecting. So just like peptide 5, tonabacet was protecting this barrier integrity. So next we did the same animal work, like I said earlier, we looked at the presence of hyperreflective foci, and we also looked at how much leakage was happening in the retina. And tonabacart was really good at protecting these. In fact, because these structures here make the whole retina thicker, so we can actually determine the effect of these structures by looking at the thickness of the retina. And we found that this thickness induced by the fluid accumulation in the retina was prevented by tonabacart treatment right across the different layers of the retina. So now we circle back to the brain. So we've done all of our experiments so far in diabetic retinopathy because it offers us a really nice way to look at the vessels in the back of the eye, and also see how the effect of treatment is. So far we've developed and characterized tonabacart, and we found that it's great and it works really well for models of diabetic retinopathy. The question here is, can it work against cognitive and ocular decline? So this work was my second postdoc, and I was very fortunate to receive the Neurological Foundation First Fellowship. And this allowed me to start to set up my own team with Ilva and Colin continuing to be my mentors. And so I collaborated with, and still collaborate with, Dr. Andrea Krakowski, who's based now in Ireland. And then I was able to recruit 2 master's students, Marvelous and Fariha, to my research team. So what was the study design? Well, we started with male 4-month-old mice, and we gave them tonabacart orally for 2 months, or vehicle, which was just water, for 2 months. And then we assessed them at different age groups, to different age points or time points. So the main the study started when they were 4 months old, like I said, and then they were given tonabacart for 2 months. And then we did the first main assessment, which we're calling the short-term assessment. And this was done immediately at the end of the tonabacart treatment to see immediately, does tonabacart have any protective effect? Then we waited 3 months, just fed and took care of the mice for the whole time. And then at 9-month-old, we did again ocular and cognitive assessments. So retinal assessments, and then we checked for their cognition or their brain function. And this we're calling the long-term assessment. So if we put this in terms of human age, we started the study, let's say in their early 20s, and then fed them right through till maybe their 30s about. And then we assess them when they're about in their 50s. So we were trying to see whether if we stop inflammation in earlier age, like earlier stages of life, can we protect against cognitive or brain damage that happens, or the brain diseases that happen later in life? To assess for cognition or brain function, we used a behavioral test known as the Novel Object Recognition Test. For this test, on day 1, you show the mice 2 objects that look the same, and then you give them about 5 minutes for them to interact with both objects. So they walk around, they sniff around them, they get familiar with both of them. Then you take them away and leave them until the next day. The next day, you change one of the objects to a new object. Now, if the mouse has good cognition, they will remember the old object, so it's no longer interesting for them. So they'll spend most of their time interacting with the new object. So this tells us that their cognition is good because they remember the last one. If they don't, they probably would spend about the same time on both objects. So this is what the Novel Object Recognition task is. And from these tasks, we can derive what's known as the discrimination index, where a higher discrimination index means they have a better cognition, and a lower one means they don't have as good cognition. The other thing we did in terms of retinal assessment was, again, the optical coherence tomography, where we took optical section through the retina, and then we measured the thickness of the different retinal layers. I won't go into too much detail on them today. So when we look at the cognition of the mice, first of all, we wanted to see how this cognition changed over time. And we saw that with increase in age, you see increase in the proportion of mice with poor cognition. Interestingly, when we look at the thickness of a certain retinal layer known as the inner nuclear layer, this was the only layer that we found decreased also with age. So then we thought, well, this is interesting. Could they be correlated to each other? So could changes in the retina actually correlate with changes in the brain? And they did. It was very interesting for us to see that, because it meant that perhaps the eye being a window to the brain could also mean that the eye could be used as a way to see what's happening in cognitive diseases, like in Alzheimer's disease, for example. So next, we wanted to see, does tonabacet slow down any of this cognitive decline? So starting with the short term, we saw that after 2 months of feeding the mice, so mice at the age of 6 months, the tonabacet treated group had a smaller proportion of poor cognition compared to the untreated group, the vehicle-treated group. But this was even more, this gap between the two was much wider when we look in the long term, because in the long term, we found that the tonabacet treatment group only decreased the proportion of mice with poor cognition by about half of what it was in the vehicle group. So tonabacet appeared to be slowing down cognitive decline in these mice compared to the vehicle. Tonabacet also protected against the changes in retinal thickness, but this was only in the long term. So in the long term, we found that the thickness of the retina in the tonabacet treated group here in green was higher than that in the untreated, in the vehicle treated group here in black, but only for the inner nuclear layer, not the other layers. So having shown this, we wanted to go back and look at clinically relevant models, because so far they've been either in vitro in cell culture or in mouse models. But as we know, a mouse model cannot truly reflect what we see in the human, does not always truly reflect what we see in the human condition. So during the Neurological Foundation Fellowship, I was fortunate to get the Health Research Council Emerging Researcher First Grant and also an AMRF project grant. And so it allowed me to expand my research team and collaborate with Professor Rinky Murphy here at the University of Auckland, get on my own research technician, Antonio, and then PhD student, Cherise. And what we wanted to see, the question we wanted to answer was, can the protective effect of tonabacet be replicated in humans? So to do this, we looked at human tissues mainly, and like I will show you later on, we're also going to look at some biomarker studies as well. So what we wanted to, the main research question here was when to treat. When does the inflammasome become involved in this disease process? And we were going back to diabetic retinopathy to answer this, because again, it's easier for us to look at the vessels. And then the second question is, are there any disease biomarkers that can be used to determine the best time to treat, or the population most likely to respond to treatment? So like I said earlier, unfortunately, current treatment targets the disease when everything is already on fire, and it's really just about keeping the damage minimum, but it doesn't affect the progression of the disease at all. Our question is, what if we can put in an alarm system that tells us that things are about to happen? Can we intervene now? And could the intervention then be tonabacet? So the first thing we did was we looked at, and this was work done by my PhD student, Cherise. We looked at control tissues where we had no known ocular pathologies and no diabetes. Then we looked at diabetes only, focusing on type 2 diabetes, where they had no known ocular pathology. And then finally, we compared to diabetic retinopathy. We wanted to know when the inflammasome was becoming involved. And interestingly, if we look at that GFAP marker, the red marker I showed you earlier, it was only increased in diabetic retinopathy to high levels, like the picture that I showed. However, when we look at the inflammasome markers, the first one being NLRP3, this marker started to actually go up in type 2 diabetes, but then was really, really high when we got to diabetic retinopathy. But then when we looked at cleaved caspase 1, which is a marker of activated inflammasome, it only really goes up in diabetic retinopathy proper when at the end of at the later stages of the disease. So when we put this on a timeline, we believe that the inflammasome is actually getting primed, getting ready to be involved in diabetes only, but then only becomes activated in diabetic retinopathy. So I like to think of this like baking a cake. So if you think of the first step to baking a cake is actually going to the grocery store and buying all the ingredients for your cake, your flour, your sugar, your eggs. That is what the priming is like. So priming is really just increasing all the proteins required for the inflammasome to go up and putting it on the benchtop, ready to go. Activation is the later stage where you then actually put everything together and you bake your cake. So activation is when the actual inflammation is triggered. So this was very interesting for us because it tells us that there is a window here where if we put in our treatment, we might be able to inhibit the inflammasome from being activated in the first place. So the next thing we did was to develop a human organotypic retinal culture model of the disease. Pretty much what this is taking a human eye and collecting little explants from it, little sections from it, and growing these sections in a plate. And then exposing these sections to our hyperglycemia and inflammation to trigger the signs of DR. So the first question is, do we actually trigger signs of DR? And the answer is yes. So when we expose that explant to this hyperglycemia and inflammation, we start to see the same increase in this GFAP red staining like we saw earlier in the confirmed DR donor. So it means that we are triggering the same environment that we will see in diabetic retinopathy. So having proved that, we then went on to put tonabacet into the treatment to see, can we reduce this red? And the answer is yes. So with tonabacet treatment, you can reduce the severity of this red staining. So reduce the level, so focusing on the red here, reduce the level of inflammation within the tissue. So this was really promising and exciting for us. So then what next? Well, next we are hoping to look at some serum-based biomarkers. So what that means is there anything in our blood that can tell us when diabetic retinopathy is coming? So for this, we're doing a study at the moment with Professor Rinke Murphy, where we have access to the blood samples from a bariatric surgery group. And this group is really interesting because they are often diabetic, and some of them already have diabetic retinopathy, but some don't. So we collect their blood over, we collect blood samples from them over a period of time, and we try to match it to people who have progressed in their diabetic retinopathy or people who have regressed, and see what in their blood, what inflammasome markers in their blood correlate with progression or not. So first of all, we'll map out all the different cytokines within their blood and see how those change with which time and across different patients, then we then do some correlation to see, for example, with patients that progress, do they have higher levels of inflammatory cytokines in their blood or not? So this will allow us to hopefully not only be able to see when best to come in to treat, but also perhaps inform clinical practice in terms of finding an easy way to diagnose diabetic retinopathy. We're also hoping in the future to look at models of Alzheimer's disease and delivering oral tonabacet to treat Alzheimer's disease. So finally, I'd like to acknowledge all of the funders that have made this research possible. So that is, with all of them are listed there. We've been very fortunate to have support from a host of different organizations here in New Zealand. I'd like to acknowledge and thank the donors that have given their tissues for research. None of what we've done will be possible without their generous donations. I'd like to thank the New Zealand National Eye Bank for their continued support. My mentors, Ilva and Colin, our Buchanan Ocular Therapeutics Unit team, and then my research team, the Inflammasome Biology Research Team, my collaborators, PhD students, technicians, master's students, optom students, summer students, everyone who's contributed to the research that I have presented today. Finally, I just wanted to conclude by saying that it's been great working with the Neurological Foundation, an organization that has slogan that really speaks to what we are about. We're really about creating a pathway to hope, because we're passionate about getting our drugs, our treatment from the bench side to the bedside, and really hoping that within the next few years, we will be able to translate some of the research that we've been doing to help patients affected by these debilitating conditions within our community. Thank you for listening.
Wow, Lola, that was just so fascinating. Thank you so much for sharing all of your work. While I was watching you go through like your whole journey of your research, I thought to myself, when did you start your master's? Like, what year did this all begin?
I started my master's in 2013, so it's almost 10 years ago now. Yeah, it's about 10 years ago. This month, actually.
Oh, happy anniversary. It is just amazing, like, to see, you know, just all of that work, that time and effort and resource, and like you mentioned at the end of your talk, all the collaborators, and, you know, over 10 years just accumulating and continuing, you know, it carries on. It's like the more you know, the more you realize you don't know.
Exactly.
And following on from that was a question that came through earlier, which I did want to highlight as well, was the phase 2 clinical trial with GSK. And I just wanted to know, was that a direct result of the work that your team had been doing with the tonabacet?
Yeah, no, that's a really
amazing
good question. Yeah, so the phase 2 clinical trial actually is not with GSK. So we took tonabacet off them, and it's with, and this is a lot of work that Professor Colin Green, like I said earlier, has been doing. And so he formed his company several years ago now. And so that company is actually heading the clinical trials. But yes, a lot of our research has contributed to that. So it's really exciting for us to see that something we've done in little old New Zealand is being, and it's being run in the clinical trials funded by the National Eye Institute, which is part of the NIH in the US. So that's really cool. Yeah.
Yeah, that is absolutely amazing. And like you say, so cool it's happening in New Zealand. And following on from that, another sort of timeline question was, when did Emeritus Professor Green developed Nabisat. I got the feeling that that was even before, way
Yes,
before.
it was before. Yes. So because I didn't want to make the presentation too complicated, so a lot of the biology I've sort of reduced, but Colin's been working on connexin blockers for decades.
Wow.
30, 40 years. And so somewhere along the way, they took on Tonabisat before I came in. And then when I joined the team, then I sort of looked at Tonabisat in inflammasome pathway and sort of tested it in diabetic retinopathy and in the neurodegenerative model I talked about. But yeah, no, it's been something going on for a long time. And so it's been really a pleasure to be part of it and to contribute my little bit to it. Yeah.
Absolutely. Yeah. It's just amazing. It's all happening here, and it's, it's a long time of a lot of work. And like I said before, a lot of resource. So it's just amazing. We've had a few questions come through from our audience, which is cool.
Yeah.
So I will just go through and have a look. Okay. This one is about the inflammasome pathway. So when does the inflammasome pathway have a good role in the body, and can blocking it cause other side effects?
Yeah, that's a very good question. So with Tonabisat, the way it works is that it blocks only a pathological channel that is upstream of the inflammasome. So it doesn't act on the inflammasome directly. It blocks another channel that feeds into the inflammasome, and that's called the connexin 43 hemichannel. Because of this, if the inflammasome is doing what it should in acute inflammation, that other channel doesn't actually become involved. So it shouldn't make you, increase your risk to anything. It's just working normally. Tonabisat only comes in and blocks it when you have chronic inflammation, which is what is bad for you. So we don't anticipate that Tonabisat will increase the risk for, or block the good part of inflammation, the acute inflammation that we want to still happen.
Okay, yes, cool. Thank you. Okay. Can I ask you, you didn't mention this in your talk, but do you know what role mast cells might play in eye disorders?
Yeah.
Curveball.
I'm trying to see. I'll need to sort of read more into it because we don't do a whopping lot on mast cells. We know that immune cells play a role. We do a lot with microglial cells, but specifically mast cells, I'm not 100% sure. But it would still be interesting to have a look at it because we know that by triggering the inflammasome pathway, we do downstream trigger, because the two main immune systems, the innate immune system and the adaptive immune system, the mast cells more part of the adaptive. So we know that eventually they become involved, but how Tonabisat fits into that, it's something that I will need to do a little bit more reading on, but I could always get back to the person if they send me an email. And yeah,
Lovely.
we can nerd out talking about it. Yeah.
Cool, awesome. So for the person that asked that question, you can flick Lola an email.
Yeah, but it's a good question.
Yeah. Okay, back to the kind of drug research trials. Does modern technology help to progress the drug research faster these days? I would say the answer is typically
not really. Unfortunately and fortunately, I guess, the approval process, which is usually by the Food and Drug Administration in the US, is pretty set. There are certain stages you need to go through. There's set things you need to tick off. So we just have to walk through the process. It's an extremely expensive process. Last I looked at the numbers, getting a drug from literally first discovery to the market is about $1 billion. So it's incredibly expensive. But you just got to walk through the process. And unfortunately, technology, I don't think has been put into that process easier yet.
Yeah, absolutely. Oh.
Mm.
$1 billion is just a remarkable number.
I know.
And think about all of the things that already are on the market, and just how much money and time's gone into that over decades of research. I've got a lot of lovely comments saying thank you so much for your presentation and how much everybody's appreciated your efforts. So I just want to reiterate that. Okay, sorry, let me just check out this question. Here we go. Wonder if the inflammasome pathway is what is involved with the reduction of asthma using non-steroidal anti-inflammatories. Do you know much about that?
Yeah, that's really interesting. So it is inflammatory still. The non-steroidals, they work on a slightly different pathway. And yes, we think that the inflammasome pathway certainly plays a role. I know work done by Professor Anthony Phillips here at the university. I remember I was part of a project with him for COVID, because we were also looking at COVID in the lungs. And so, yes, no, certainly the inflammasome is involved, and we have other projects around looking at whether we can actually make tonabacet into kind of an inhaler type of thing to treat lung-based inflammation. But I'm not too directly involved with that, but I can certainly send whoever it is down the path of who is, if they wanted to know more.
Yeah, amazing. Okay, what would be the better way of the administration for tonabacet for treating Alzheimer's, or has there been any research into tonabacet in those diseases or Parkinson's?
Yeah, well, that's the next step for me. So I'm developing a research program to look at tonabacet for Alzheimer's disease. And we're hoping to start off orally, because that's what we would prefer. The great thing with tonabacet is that it crosses the blood-brain barrier. So we don't need to worry about getting it directly into the brain. It can cross the blood vessels into the brain anyway.
Amazing.
So we will try it as an oral drug in the first instance. It's still several years away, but yeah, we just keep soldiering on, and hopefully it works, and we'll see. But Parkinson's disease, the problem is the models. I've not seen any good models for Parkinson's just yet, but it doesn't mean that we can't try. But yeah, at the moment we're just doing Alzheimer's first, but yeah, certainly in the future. These diseases, Alzheimer's, Parkinson's, even diabetic retinopathy, they all seem to share the same overarching mechanism, in that the inflammasome is involved. So we would anticipate that if it works for any of them, for Alzheimer's, it should work for Parkinson's. But yeah, it will be something in the future for sure.
Amazing. I look forward to hearing about that research down the track. And like you say, we soldier on that pathway to hope.
Yeah, exactly. That's exactly what it is.
And sort of following on from that one, do you know, I guess you may find this out in your future research, if you know the age that the inflammasome pathway might start causing issues in Alzheimer's?
Yeah, so that's the first question we want to answer. So that's the very first question we're hoping to answer. So we're collaborating in the near future to get some tissues that allows us to map out or profile the inflammasome over different ages, and put on top of that profile the cognitive decline. So we can see how the cognitive decline progresses with age, and how the inflammasome comes through with age. And then we can pick, okay, so this is when the inflammasome comes in, can we treat then to then hopefully reduce that cognitive decline? So that's literally our first question. And then once we've answered that, then we'll put tonabacet into the picture. And
see the effects of it there.
Yes.
Just another comment. Thank you for a very interesting and hope-filled presentation. So your work is very appreciated out there.
Oh, that's great. That's really good. I'm glad.
Yeah. We've got time for one more question. So I just thought this one just came through on the back of the Alzheimer's chat. could investigating the eye provide a window into Alzheimer's disease? I suppose the eye is the window to the brain.
Yeah, again, it's part of the project.
Oh, can't wait for your
It
new
is part of
project.
the next program. I've got an amazing PhD student, Amy, working with me at the moment, and she's actually already doing that. So she's looking at the eyes of a model with Alzheimer's disease. So she's doing the behavioral, and at the same time, she's doing the ocular assessments. And I mean, the results are only just coming in, so we don't know just yet. We're still analyzing, but we would think that the eye will be involved as well. Because even in patients with Alzheimer's disease, we know that unfortunately they do sometimes have ocular, retinal defects as well.
Yeah.
And some studies suggest that you can actually have some of that amyloid beta, which is the main thing in Alzheimer's disease, also depositing in the retina. So,
oh, wow.
We would anticipate that we will have ocular effects too. So hopefully by targeting upstream and targeting the inflammasome, we could do a double whammy and treat both the eyes and the brain at the same time, which is so cool.
Yeah, wouldn't that be a dream?
Yeah, that's what we're hoping for.
Awesome. Well, that's all the time that we have for today. But Lola, I just want to say thank you so much again. And everybody, I can see from the comments coming through, just really enjoyed your talk and how you made it so accessible for everyone to understand and get a bit more information about the eye. Thanks to everybody at home for joining us. As I said earlier, this event, the recording will be available on our website later this week. And thank you for coming to any and all of our Brain Awareness Month events this month. So thanks everyone, and we will see you again soon. Thanks, Lola.
Thank you.
Ka kite.
Bye.